think it's time to get started and welcome you all to the next installment of the GHGA webinar, this time covering interpretation of results from biostatistics with Sarah Kaspar from the Center for Statistical Data Analysis from EMBL. And if you are curious about any of our other webinars, I would like to invite you to go to ghga.de/resources/training. There you'll find a lot of material that we covered in our previous webinars, as well as our YouTube playlist where all of the videos from the webinars are available to you. And now I would like to introduce our speaker today, Sarah, who got her bachelor's, her master's, and her PhD in both molecular biotechnology and biology from the University of Heidelberg. And since 2020 has been working as a biostatistician who leads the Center of Statistical Data Analysis at EMBL. So she does a lot of statistical training, she offers consulting, and today she's going to talk to us about some of her work. And with that, Sarah, the stage is yours.
Yeah, then also hello everyone from my side. Thank you for the introduction, and also thank you for inviting me. So as you said, I'm going to talk about concepts in biological hypothesis testing today, with a focus on how to interpret results from publications. So it's really an introductory lesson, but I will link to further materials in one of the last slides of this presentation. And I think Julia is about to share the slides with you in the chat. Thank you for that. Okay. So since you all joined the GHGA webinar, I assume that you're somewhat interested in biological data. And you're also aware that we live in times where we have an increasing amount of such data, right? We have several omics techniques available. In addition to that, we have imaging techniques, we have clinical data, and sometimes these techniques are also combined in the same studies. So the example that I brought today is on transcriptomics. We will be dealing with a 20,000 over 32 matrix and metadata. That's rather small. But still, it turns out that human brains are not particularly good at dealing with these kind of matrices. So what we need is ways to make the data accessible to our human brains. And also, in the end of the day, we will want to trust somehow the results that we look at. So in addition to there being lots of data, they also often have come a long way. So as an example, you see here the workflow of RNA sequencing. There are several experimental steps followed by some in situ on the computer steps. And somehow statistics, though debatable, is often seen as the end of that pipeline. Because if you read a statement that says there's a difference between the conditions, is not trusted unless it reads there is a significant difference between the conditions and it has some p-value on it. So as a general biostatistician, I see both the small and the large data sets from researchers, biological researchers, and I also hear a lot of doubts. And I just wanted to share with you some of the questions or doubts that I hear on a daily basis in my work. So why is it the p-value that we trust in, and should it be that? Why does everything in stats have such clumsy definitions? Why do the results seem to depend on which particular statistician we asked? And finally, can't we just go and interpret anything into big data sets? So if these are also your questions, I promise I'll do my best today to address all of them. But before we start, I also wanted to point that statistics is about more than just p-values. It's also about p-values, but it's also, to begin with, about visualizing data, estimating biologically relevant quantities, yes, testing hypotheses, but also finding patterns in the data, especially when we're talking about large data. So one thing that a lot of these methods or statistical methods in general have in common is that they try to reduce the complexity of the data. So for example, we do principal component analysis to bring high dimensional data sets down to 2 dimensions so we can look at them. we often report averages, right? So for example, if we have a genetic region, we might report the average methylation levels, not from each sample. Also, if we do a hypothesis test and we have a p-value, that's one value that comes out of a big data set. And finally, if we do RNA-seq or transcriptomics, we start with a huge number of genes and we end up with a candidate list of genes that we might look into further then. Now, reducing complexity is of course helpful, but it also means that we're losing information. So what you see here is the famous Anscombe quartet. That's four small data sets that each plot two variables against each other, X and Y. And I think it's relatively clear that these are four quite different scenarios that you see here. However, if you look at certain summaries of the data, for example, the average in X and Y, also the line that we might fit or the standard deviation, they are all the same in all of these four data sets. So this means that we should, we can use summaries of the data, but we should be aware that we cannot blindly trust them. But we should also, and this is already the first lesson for today, try to get a look at the data. It sounds trivial, but is very helpful in the end. And often people seem to overlook that step. All right, so what I've just shown you with the mean and the standard deviation and the correlation, these were all so-called summary statistics. So that's one value that describes the sample that you have at hand. But what we often want to do in the end is we want to draw some conclusion on a population or on a bigger population of interest based on a sample. So let's say if we have 10 bags of apples and we count the number of worms in each of these bags, then we could describe these bags of apples with some average number of worms in them or something. But maybe what we want to do is make a statement about all the apples from one brand or even all the apples in the world and how many worms they have in them. So what we do is we sample, we generate data from our population of interest, and then we use some methods and try to make inference, so try to make a statement about what we are really interested in, this population of interest. This always involves some assumptions. And these assumptions often, but not always, come in the form of a statistical model. So this is, I think, a very useful topic to look into. So here the idea is that we have some data generating process. So for example, the way in which you choose your 10 bags of apples, and then maybe you have some complicated machine that slices them and makes some heuristic about how many worms you have in each bag and spits out some count. That's a data generating process. And now the model is our view of that process, which is why I try to picture it with these pairs of glasses, right? So the model tries to represent the data generating process. And it does so by summarizing a set of assumptions, often often always actually in the form of equations, but I promise to spare you with equations today. So a set of assumptions could be, for example, that all the bags that you have the same size. And you could also specify relations between the variables. For example, you could say that the age of the apples increases the number of worms that you'd expect with them. And it also simplifies the data generating process a bit. Yeah? So your splicing counting machine may be a little bit less complex than it is in reality. And what we use the model for in the end is we use it to calculate the probability of some data that we might see under these assumptions that we make in the model. And I will try to be a little bit more specific about this with another example. So let's say we have some disease and it has a known prevalence of 4%. That means 4% of the whole population have this kind of disease. And now we're interested in whether a precondition somehow changes the chances of getting this disease. So the precondition might be diabetes. So what we do is we get 100 test persons that have diabetes and test them for the disease. And out of these 100, we see 9 that have the disease. Okay, is that surprising? Is it not? We don't know unless we set up some kind of model. And here the binomial model is very easy and very useful, I would say. In the binomial model, we can take this prevalence of 4% as our model. And in this model just says that every test person has a probability of 4% of getting the disease. And then in this probability distribution, you see the probabilities of seeing 0, 1, 2, 3, and so on test persons out of the diabetes group that have the disease. So we expect around 4 out of 100, because it's 4% in our model. What we see is 9. And here, finally, you can see that the probability of having such a high value is really small, starting from this assumption of 4%. So we might decide that maybe the prevalence in the diabetes group is higher or different in the end. Okay. So I guess a lot of you have already heard about this famous quote from George Box, which says, all models are wrong, but some are useful. And I think it quite nicely sums up the daily struggles that we have in statistics when we ask ourselves whether a particular model is applicable to the data set or not. So for this example, why is it wrong, this model, and why is it useful? So it's wrong because it's oversimplifying. For sure it is, right? So factors like age, the region where participants live, or other preconditions are completely neglected. And for sure, our test group of 100 persons is not completely representative of the whole population. Also, where does this 4% come from, right? We assume 4%, and it has also been measured, hopefully, at some point. Also on some group that was for sure not 100% representative for the population. So these 4% here are also subject to uncertainty. So we could now trash the model and go home, or we could decide that it's still a useful model for this case because it gives us a straightforward estimate of how unusual the data are under these assumptions. And this can be a starting point for further investigation. So here on this 9 out of 100 data set, we can, for example, learn using this binomial model that when the true prevalence is 4%, the probability of observing an event of 9 out of 100 or more extreme is just 2%. So this 2% is a p-value, right? And it's rather low. And having this low p-value can, as I said, be a starting point where we can decide based on that there might be something interesting with diabetes and this disease that we're looking at, and we can further investigate. Okay, so now finally we are going to look into real data from a publication. And in this, the authors were looking for pathways that are associated with the outcome of an RSV challenge. So RSV is a respiratory syncytial virus that small kids often get, and they get very sick. So they had a group of adult volunteers. Yeah. From them, they got nasal swabs, which went to RNA sequencing. So they had the gene expression profile from the nasal tissue for each test or for each of these volunteers. Then they challenged each of the volunteers with the virus, so that I think they put some drops of it in the nose. And then after a couple of days, some of the participants got a cold and some didn't. And now that the group divided into two groups, the question was, of course, whether there were genes that are differentially expressed in the cold group before exposure. And maybe they are somehow showing pathways that are predictive for getting the disease or not. Okay, so what we have is a sample out of each test person. And in a nutshell, the samples go into some procedure where the RNA is extracted and fragmented, prepared for sequence. And then from the sequencing reads, they are on the computer mapped to some reference genome. And this leaves us here with expression data that looks as follows. Here we have a matrix that gives us for each sample and each gene the number of reads that map to that gene in that sample. So we have about 20,000 protein coding genes in humans, and we have 32 samples in that particular experiment. So we have this count table, and we have also some metadata that's telling us for each sample, for example, whether it belonged to an individual that got a cold or not. What we want to do is we go through the genes, right? And we want to know for a particular gene, whether the expression in the cold group was higher or lower or the same as in the no cold group. And that's the setup. And we can just start by looking at a more or less random gene from, not totally random, but a bit from the genes that they tested. It's called MXD1, and it codes for a transcription factor that mediates cellular proliferation and other processes. So in this plot, I'm showing you normalized counts. They are normalized to the sample size. And we have 16 counts from 16 persons in the no cold group and 16 counts from the cold group. I'm also showing you the log averages in each group here as red points. And I mean, you clearly see that the normalized counts differ in their log averages, but well, we know it's biology, and we know there is lots of variation between individuals. So this difference could be by chance. And we'd like to know how likely it is to see such a difference just by chance, if in reality the expression of MXD1 had nothing to do with the response to the viral exposure. So how do we calculate probabilities? Because just by chance is a probability in the end, right? We use a statistical model, and some very common model for gene expression goes as follows. We say that the expression of the gene is described by some baseline expression. Baseline in this case for the no cold group. Then there is some difference between means, between average expression between the cold and the no cold group. This could be 0. This is what we're about to test. We have some typical variance that we expect for count procedures. And we have overdispersion, which is additional variance that is due to biological or also technical heterogeneity, because after all, we are dealing with biological samples where we usually see lots of heterogeneity. So since I said this difference between means could be 0 or not, whether it is or not defines two models that belong to two hypotheses. So in hypothesis test, we have the null and the alternative hypothesis. And in this case here, they can be described by a null model and an alternative model. I try to picture it again with the classes. So the null model has the classes that are both at the same level, and it says that there is no difference between the groups. The alternative model says that the groups are different. So there are two different means. So how do we decide which model we believe in the end? So what we do is we start by assuming that the null model is true unless we have enough evidence against it. And I try to illustrate that a little bit again. So you can now imagine me in 20 years. That's me in 20 years, I will be super grumpy. I think that new discoveries are completely overrated, which means I wear my null hypothesis glasses, and I will believe that there is no difference between the two groups in your data, which you are showing to me, unless I have enough evidence against this view. And evidence for me means that the probability of the data that you're showing under my view, under my null model, is low. And here is how in practice we usually calculate this probability. So what we do is we summarize the data into a test statistic. And in this particular example, it's called Z. And this test statistic should be large when the data deviate a lot from the null model. Usually this is the case when we have low variance within each of the groups. when we have a large sample size, and if we have a large effect size, that means a large difference between the means. So in the end, this test statistic Z weighs the effect size and the sample size against the variance. Then we compare this Z that we got from our data set to a null distribution. And this null distribution gives the expectation of Z under the null model. This null model comes either from mathematicians that derived it, or you could also simulate your own null distribution if you wanted to, right? If you have a null model, you can simulate data from that null model again and again and see how the Z looks. And then you will get some kind of distribution. Finally, we have the p-value, which gives the probability under the null to see a value of Z at least as extreme as the one that we observed from the data. And like geometrically speaking, we are comparing these tails of the distribution here in blue, where the outcome is more extreme than what we have to the whole area of the distribution. And then we can make a decision and we can reject the null hypothesis if the p-value is below some significance level alpha. And here convention is 5%, but this is like completely arbitrary. You could also pick your own alpha if you wanted to. Okay, if we apply this procedure to the gene that I'm showing you here, we would get a p-value that is like really low, 0.00013. So our conclusion would look something like that. We could say the gene MXD1 is differentially expressed between the cold and the no cold group with a very low p-value. And this brings me to the second lesson, which is really my favorite intuition for the p-value. The p-value quantifies evidence against the null hypothesis. This is how I think of the p-value. And now that we have some understanding of the p-value, we can skip a little bit ahead in the publication. So they tested, of course, not only this one gene, but all of the 20,000 genes, and they got a list of genes where the difference between the groups was significant. And then they looked into this list and said they found a biological process associated with neutrophil activation in which unusually many of these genes from the candidate list were involved in. So that's some kind of new hypothesis. So maybe the ones, the group that got the cold in the end had higher neutrophil activation, which is already some kind of immune response going on. And if you have a hypothesis from such kind of a genetic screen, a transcriptomic screen, what you can do is follow-up experiments. And we will look at one of these. So now what they did, they tested nasal protein levels to test their hypothesis. Of some proteins that are indicative of neutrophil activation, and here it reads that the nasal proteins were significantly higher in the cold group relative to the no cold group, and a p-value. And now that you learned a little bit where the p-value come from, let's start from such a statement and dissect it a little bit, and let's see what we can say about this and what else we should ask for. Right? So the definition here, or the translation of this statement is under the assumption that the test makes, that they used, and under the assumption that there is no difference between the protein levels in the two groups, the probability of seeing this kind of data is 0.01. So if you were wondering why results seem to depend sometimes on which particular statistician we ask, then the answer is partly here in the assumptions that the test makes, because here you can always debate. So if you want to argue about some statistical results, of course, you can challenge the assumptions and discuss about them. Also, it might depend on how exactly you phrase your research questions. So this is, from my experience, what I spend a lot of time on, discussing with researchers what their actual question is and what the according test that we should do should be in the end. And then there was these clumsy definitions, right? Why is this such a long sentence? And couldn't we just say that the p-value is the probability that the null hypothesis is true? So the problem here is, if the p-value was the probability of the null hypothesis being true, we could in the end prove anything by just collecting very little data, right? Because remember, we start by assuming that there's nothing to see in the data and the null hypothesis is true. And then just if you provide enough evidence, grumpy me will be convinced that the null hypothesis can be rejected. So you could just go and show like no data points at all. You will not convince anyone that the null hypothesis is false. And then you say like, I knew it, I proved the null. So, I mean, this is a little bit exaggerated, but in the end, that's how it is. Little evidence against the null hypothesis doesn't mean that the null hypothesis is true. So unfortunately, for now, we'll have to stick with these kind of clumsy definitions. Yeah, why is it the p-value that we should trust in, and should it really be the p-value? So I hope I convinced you that the p-value is a useful quantity to get out of your data, because it's evidence against the null. But in fact, the p-value is some kind of a summary of the data that consists of different things, including variability of the data, sample size, and effect size. And that means your p-value can also get small due to different reasons. So here I'm showing you 3 different toy data sets. There are comparisons between hypothetic no-cold and cold group, which all have the same very low p-value. So in one example, the p-value is low because there's really a substantial difference in expression between the 2 groups. In one example, there's rather little difference in expression, but we have lots of data points resolving that small difference. So again, lots of evidence. And in the last example, there is hardly any difference, but also the variance is basically not existent. So this is why we would also call this difference significant in the end. And what you should do if you see a p-value is therefore, you should look at the effect size, right? You should also look at the difference in means that you see. Because also remember how the null and the alternative hypothesis are defined, right? We have the null hypothesis that says there's no fold change between the groups or no difference between the groups in expression or in protein levels. And the alternative hypothesis is really anything else. This means if we had the tiniest change in expression or protein level, and we had just enough data points to resolve it, this would be enough to reject the null. And now I'm showing you here the transcription and regulatory network for E. coli, where you see that basically everything is connected somehow with everything. So the question is, would you really expect that if something changes the expression of some of these genes, that this change causes no change in expression at all for the other genes? Well, probably it does, but the differences are tiny, and we're likely not interested in this kind of difference. But what we're interested in is the genes that have a biologically relevant expression, some kind of substantial expression change, not a statistically significant one. So I repeat my point, have a look at the effect size. So in that particular paper, we don't get an effect size reported, but we have the chance to look at the data, which is also good. So here you see the MPO protein levels. And it's also clear to the eye, I would say, that in the cold group, the protein levels are almost consistently higher, and I would guess here is maybe a fold change of 5. So I can live with that. So finally, this is a big question, right? Can we just go and interpret anything into big data sets? And yes, I see that there is some danger to that, but also there are good practices that should keep us from doing so. For example, if the data sets are published, right, someone can look it up, and if you describe the results in a way that they are reproducible, others can try to reproduce the results. So there's one step of quality control, and there are also other types of quality control that are suitable for big data sets. I'm going to highlight one of them, which is adjustment for multiple testing. Right, so here in the paper they found a list of 80 differentially expressed genes whose p-value was below some cutoff. But here they don't write the p-value is below 5%, but the adjusted p-value is below 5%. So what's the difference between those? Well, why do we need some kind of adjustment? Okay, so for this, I would like to come back to this hypothesis test that I showed you in the beginning, where we had a group of 100 test persons. We wanted to know whether the disease prevalence in this group is somehow different from 4%. And we had here 9 persons with the disease, and we did some binomial model and binomial test, and this came out significant with a low p-value. So this model is now so easy and so simple that we can also simulate this kind of experiment by saying we take 100 persons, each of these has a disease or a chance of getting the disease of 4%, and we do this 200 times. So 200 times I simulate a data set out of 100 persons. And these are the counts that we get here, right? So most of the time we would expect between like 2 and 6 persons with that disease out of this kind of data. But sometimes we also see more extreme values just by chance. And here also in light blue, the significance test comes out, well, significant. And these are false positives, right? I simulated the data myself. There's nothing going on, but sometimes the test, well, comes out false positive. So that means when you think about it, when we're testing at a significance level of 5% and the null hypothesis is true, then by definition of the significance level, we expect to see 5 out of 100 tests to be false positive, to still be significant. And the more tests we perform, also the more false positives we should expect. And this means that we need some kind of additional control to make sure that we found more hits than we expected just by chance. So I'm not going to go into the details of the methods. As I said, I will link resources for this if you want to look further into this. But for a large data set with 20,000 genes, if in this case they had reported all the genes with the p-value below 5%, then the list of candidate genes would contain 1,590 genes, of which 44% would be expected to be false positive. This I looked up in the data earlier on. And what you can do in this case is just use a stricter p-value cutoff. And by this, they ended up with this list of 80 genes, of which they can expect 5% false positive, so roughly 4. Unfortunately, we don't know which ones these are, because otherwise we could delete them and we would have no false positives. Yeah, so this is just for you to know that this problem of false positive, yeah, is a problem, but we have ways to deal with that. Okay. So now that I've answered lots of questions that you never asked, I thought in the end of this talk, I would also give you some new questions that you can ask yourself or your collaborators when you look at statistical results. So does the test answer the scientific question, right? We talked about this. It sounds easy, but sometimes it's not. If you want to argue or challenge the test, you can also ask whether the assumptions that are made in the test are somehow reasonable. It's always good if you can have a look at the data points themselves. And if you have something that is a significant result, you should also ask yourself if it's a biologically relevant result or if it's just a tiny effect that has resolved by many data points or due to low variance. Finally, if you're looking at large data sets, you should ask whether there was a correction for multiple testing, and if there was not, then you should ask whether there was a rationale behind performing exactly the test that you see in the publication. Because, right, if you have a big data set, you could do lots of tests, and yeah, I think sometimes it's easy to cheat by showing just the tests where something was found. All right, I have here also the list for further reading that you can look up from the link of this presentation in the chat. But for now, I have said everything. Thank you for listening, and I'm open for any questions.